Hi, friends of Sun. Welcome to our reading group. We have this thing here happening every week, and you can join us every Monday on Zoom if you want, ask questions yourself, and so on. And yeah, I guess we always discuss a paper here that I kind of find interesting and invite the authors to discuss their paper with us or present it. And today it's some DNA diffusion stuff and trying to generate regular DNA. So let's go.
My name is Peter Koo. I'm an assistant professor at Cold Spring Harbor Laboratory. I run a research group focused on deep learning applications in genomics with an emphasis on interpretable AI.
Yeah. Hi, I'm Anirban. I completed my PhD a couple of years back, and then I spent 1 year at MIT as a postdoc, and then I joined Peter's lab. So, yeah.
Okay, should we just get started then?
Yep.
Sounds good. So today we'll be presenting recent work from my lab on designing DNA with tunable regulatory activity using discrete diffusion. Okay, now I'll start by providing some biological motivation. So we all know that proteins are the workforces in a cell, carrying out most cellular functions, but they only comprise 1% of the genome. And a significant portion of the non-coding genome controls gene regulation, which determines protein expression levels. And these levels vary quite a bit across different tissues in the body. Since many disease-associated variants fall in this non-coding region of the genome, studying how they drive the dysregulation of protein expression levels is an important problem for human health. Now, diving a bit deeper into the non-coding genome, transcriptional regulation requires the complex coordination of many, many proteins. And in this simplified diagram, it shows that proteins are interacting with DNA at cis-regulatory elements, which we often refer to as CREs. And these CREs are DNA sequence elements such as enhancers and promoters. And within the CREs, short sequence elements called motifs coordinate the binding of transcription factors to regulate gene expression. And the rules governing this coordination are known as the cis-regulatory code. And decoding this complex language of the regulatory genome, i.e. the cis-regulatory code, is a major goal in genomics. High-throughput functional genomics experiments have produced extensive datasets linking DNA sequences to their molecular functions. And these functions typically include protein binding, enhancer activity, gene expression levels, and so on. And so training deep learning models to take DNA sequences as input and directly predict the functional readout of experiments in a supervised manner is the most widespread approach to modeling these types of regulatory genomics data.
Peter, what sort of is the thing that we're predicting here, right? The piece of DNA that you have here, which if it's maybe an enhancer, then it'll also be cell type specific and maybe its activity is specific to also a certain protein in the cell, right? It can enhance a protein and depending on what cell it is in, it can enhance a protein. So are we making these biochemical activity predictions with respect to a certain protein or is this just a technical thing?
So exactly, those are very important points, and I was going to highlight some of the limitations because of the cell type specificity. In these cases, these are cell type specific models. So when we train them on just DNA sequences and predicting protein binding or the enhancer's activity, it's only true within that cell type. And these supervised models have to make these assumptions because there's a confounding factor of the state of the cell. And as the state of the cell changes, the expression levels of the proteins vary too, and that changes the affinities of binding to these sequences as well. So we assume that everything else is constant all the time with these models.
Yeah, okay, but is the prediction with respect to now a single protein? Like, is the prediction?
I see what you're saying. So it could be. It depends. So if it's a certain type of data set, like a ChIP-seq data set, then it's with respect to a single protein within a single cell type. But we can also do DNA sequences over longer regions and predict enhancer activity. Or even like our DNA sequences can be at the length of hundreds of kilobases, and we can have our models predict gene expression levels, like mRNA-seq, for instance.
Okay. Yeah.
So I was just trying to group them all sort of in a big bag and just put them all together because they all have the same theme.
We have these different types of assays: ChIP-seq, ATAC-seq, mRNA-seq also.
Yeah.
And with all of those, we get different data, which is either target protein specific or not target protein specific. And that's then also what we train our models with, I suppose.
Yeah.
Good, good.
Okay. So, these supervised models have been incredibly transformative for fitting and interpreting these functional genomics data sets. For instance, we can use a high-performing deep neural network, or DNN, to score the functional consequences of non-coding variants. And this can help us to prioritize the thousands of disease-associated variants identified from genome-wide association studies. Through post-hoc explanations, such as attribution analysis or counterfactual explanations, our field has repeatedly demonstrated that genomic deep learning models indeed learn the locations of protein binding sites, their binding strengths, as well as higher order dependencies. And these insights have been consistently validated in different studies, revealing the strength of this class of discriminative models. And we can also use the sequence of function deep neural networks to design novel sequences with desirable properties. Sequences designed in this manner via sampling a discriminative model have been experimentally validated and offer a promising direction for the field. And so I'll give you one intriguing example of a downstream application for this sort of designing task. Suppose we want to deliver a payload like an mRNA into a specific tissue as part of a drug therapy. We can use a black box optimization with these supervised models to design a regulatory sequence that only transcribes that payload inside the cell type of interest, thereby reducing any off-target effects. Now, while these supervised models have been proven to be quite powerful, they have also serious limitations. For instance, they have been found to generalize poorly under covariate shifts. It's been shown that state-of-the-art models right now that we have can give incorrect predictions when introducing just a few mutations, such as population-level genetic variability. And second, discriminative learning doesn't require a full understanding of the cis-regulatory codes. It can learn just what it needs to make the right predictions. So it may miss the full spectrum of patterns simply because it's focused on prediction task rather than learning the joint data distribution. And third, these discriminative models are known to easily exploit shortcuts in the data. And in genomics, we have lots of shortcuts that we don't even understand fully. So on the other hand, generative modeling has not yet been proven for regulatory genomics. And this is a research topic that I've actually personally struggled with from my days as a postdoc. And the struggles have continued as a PI with numerous failed rotation projects and summer student projects. And I feel pretty bad for all the lives I've destroyed by putting students on generative modeling projects for regulatory genomics. And, jokes aside, what makes this frustrating is that approaches like DAEs, GANs, and large language models work pretty well out of the box for protein sequences, but they face significant challenges when applied to regulatory DNA. And so generative modeling in genomics have greatly lagged behind their protein counterparts. And I'll explain why. So this is largely due to fundamental differences between proteins and non-coding DNA. Proteins possess natural start and endpoints and exhibit conservation at both sequence and covariation levels, which reflects their structural properties. In contrast, non-coding genomes are much more complex. They're characterized by high variability in their positions, their lengths, and composition. And this is because evolutionary constraints are not imposed at the sequence level, but rather at the functional level. Now, here's an important nugget of insight. Diverse arrangements of strong and weak motifs can produce the same functional outcome, which means that regulatory DNA can drift quite a bit while still maintaining function. So they are actually quite diverged across species. Second, the challenges of modeling non-coding DNA are further compounded by a significant class imbalance. Important regulatory sites are relatively sparse and scattered among seemingly random nucleotides. So when training generative models using reconstruction-based losses or even language modeling objectives, this extreme class imbalance of information means that at most positions, the model is tasked with predicting uninformative positions, which by definition can only occur via memorization. And third, the relationship between DNA sequence and cellular behavior is highly complex, as you were mentioning earlier, Hannes. While every cell in a multicellular organism shares the same exact DNA, or roughly the same DNA, cell types exhibit widely varying behaviors due to differential regulation encoded in the genome. And this encoding is very complex and activated based on spatial temporal cues. So generative models, including language models that focus solely on DNA for their training, are unlikely to capture the full complexity of cell type-specific cis-regulatory codes in regulatory DNA.
Okay. So recently, diffusion models have emerged as a promising approach for deep generative modeling of DNA sequences. These approaches span standard denoising diffusion probabilistic models, latent diffusion to modified diffusion models that better accommodate the categorical nature of DNA sequences. And the latest work in this direction is digital flow matching, which is led by Hans. However, the evaluation of these models have been on the lighter side, leaving open questions of whether these models are a significant advance in the generative modeling of regulatory DNA. Here we're going to think in terms of diffusion with score matching, which has been very successful for images. For standard score matching, we train a deep network to approximate it to output the ground truth score. But this score can only be calculated for continuous distribution. In our specific case, the data inherently follows discrete distribution, and in this work, we follow a discrete version of score matching that seemed to have worked nicely for text modeling. And this is the outline of today's presentation, where we talk about the methodology in the first half, followed by the evaluation framework in the second half.
Okay, so like we now had a sort of motivation. Why do we want to generate DNA sequences? And I guess this is kind of because we have some theory, or one reason is that we just want to understand how nature works, and another reason is we want to generate maybe some new enhancers, for example, in therapeutic applications. And Peter, for example, you made this you made this example of having a drug that we only want to be active in a certain cell type, and then we put some RNA into a cell, and then we design this RNA, put the RNA into a cell, but it also has some enhancer attached to it or so, and then or some promoter or enhancer attached to it, and that then means it's only is active in that particular type of cell where we want it to be active. Right.
Yeah.
Okay. And now we want to be able to generate these enhancers that are only active in a certain type of cell. For example, we have a discrete sequence of stuff that we want to generate, and that's why you'll now explain why we are interested in some, now you'll explain how you generate discrete sequences, like with your with sets. I guess. Yeah.
Yeah. Yeah. Perfect.
Okay. Okay. So given a discrete sequence data X following distribution Q with finite cardinality S, diffusion is just an evolution of Qt given by the ordinary differential equation dQt is equal to RTQT. Which is driven by a transition rate matrix RT with time steps T. And we'll see more about this transition rate matrix RT later. And this transition rate matrix RT controls how often one state goes to other states. And the transitions are given by this equation here for a small Euler step delta T.
Okay. Let's maybe go. I mean, this is a bit quick. So we're saying we are at state xt right now, and then we looking here at the probability of going to xt or of going to xj at the next time. And then we have this delta here, which says that this probability is
so this delta here is suggests that how long the step can be, and this is standard Euler step, and
no, I mean your small delta, your delta ij.
Oh yeah, so that is a Kronecker delta, and this is just a very small value, just to avoid this numerical instability.
Okay, so is it okay? So in this RT from of going to J to I, there we have the this is a rate matrix. So
Right.
it's not, but so it's not directly the probability of going from I to J. It is the rate which we multiply with the time difference.
Yeah. So I mean, in the next slide, I'm going to show an example. So probably it'll be more.
Let's do it. Let's go to the sum.
Yeah. So the first example contains a standard rate matrix, which can be one particular. I mean, so this one example of transition rate matrix. This matrix is required to follow a specific structure so that when they are applied to transform a distribution Q, the rates should sum to 0, and this ensures that the data at every intermediate state follow a distribution. So this is just one example, and following the previous ordinary differential equations shown above, or shown in the previous slide, or the previous slide. One solution of Qt can be exponentiation of T multiplied by the rate matrix. So this can be one example of the rate matrix for this case. And this is just one example of Q0, and yeah, so.
Can you talk a little bit about why should we interpret this as a rate? Like this is now the rate of going. Let's look at the 0, 0 entry. Why is this the rate of going from state 0 to state 0?
Okay, so this is very similar to in standard diffusion model. You may consider adding Gaussian noise. But here, this is kind of similar. It's like you consider applying some particular transition that allows you to move from one state to another. And
so it's clear. I'm just asking about the interpretation of this matrix. Why is the 0, 0th entry a rate? Like -3. What's the rate of -3?
Oh yeah, so this is yeah. I mean, you were right. So this is just one example, and the standard that the actual matrix that we are going to use is not this one.
Sure. Okay. But then in this example, what is like how can I interpret this -3 in a rate, right? We have this probability distribution.
It's exponential. There is an exponential, so it's a probability, something like this.
That's not helpful to me.
Yeah. This is, I mean, so this particular rate matrix should be considered in such a way that that if you multiply with all the columns should sum to 0, so that when you multiply this to the distribution values, this masses, it should give you something which should, which should be the rates, and those rates should sum to 0. And these values can be anything, and it is just an example. So
Yeah. Okay. Anyway, let's continue.
Okay. I mean, we'll see examples, more examples later.
Yeah.
Okay. So in general, RT can be analytically chosen as RT equal to beta T RB, where beta T is a time-dependent scalar and RB is time-independent base rate matrix RB. In this scenario, the continuous time Markov chain needs to be integrated to obtain QT as exponential beta T RB beta T bar RBQ0, where beta T bar is accumulation of beta T until timestamp T. Specifically, beta is a noise level where beta T bar is the cumulative noise. And there are many methods to compute matrix exponential, such as eigenvalues, but we consider a simpler one, and this allows us to calculate the transition of the true sequences for time t, for any time step t. Also, RT should be considered in such a way that QT tends to a terminal distribution for large T. Okay. So we'll talk about concrete score here, as score function can be calculated in discrete space. The quantity Qi over Qx, that is the density ratios, can be considered as the categorical equivalent of the famous score function, which is basically a vector field pointing to the direction of higher probability mass. And now we can write out our neural network approximation as S theta of x, emphasizing that a single network evaluation should evaluate all ratios with denominator Q of x. Modeling that would have n power d items, which is computationally intractable. Hence, we only model ratios based on whether y is close to x. We consider all sequences that differ from x at one position, that is one Hamming distance away, which reduces the complexity drastically. Here we show an example of possible neighbors for a given text.
Okay. Then this was a bit quick for me. Can we go back a slide?
Yeah.
Or maybe one more, or what came before this slide?
Yeah.
Okay. No, I guess then. Right. Here we're okay. Here we're writing down one way to get the transition matrix, I guess, from the rate matrix. And then if you go to the next slide, you're saying okay. So the difference between the value of two points, if you evaluate your function at 2 points, the difference of that.
So yeah, I mean, basically this is can be considered an approximation of the true score function, which we can calculate for continuous.
Okay, so is it basically that you look around at all of your neighbors or something like that, and then if you take the difference in values of the difference, yeah. Okay, we want to approximate the score at a certain point, and now to do that, we write the score as the gradient of the log density. And so now we look at the density that's all around us, at the density that is where we the density where we currently are, and then yeah, the density where we currently are, and then we like why is it the can you explain a little bit the connection of first line, second line, right? If we take logs, then minus becomes then becomes
so basically this log this grad of log qx we can write as, I mean, grad qx given over qx. And the way this score function works is like it it is basically a vector field pointing to higher density regions. But in the discrete space, we can consider that, I mean, if you are at some probability mass for some point in your discrete space, you can think about that this analogous to this score analogous for discrete space can give you the directions for higher mass regions in that space. And here you can think about think about many possibilities. I mean, there are maybe many neighbors, but to reduce the complexity, we are only considering neighbors with one Hamming distance. So which are
just wait a second. So can we also talk about this in a continuous setting if we just say we can do all integrals that we want to do? Then right, it's clear how we go from the grad log Q to grad Q over Q. But then this approximation here, what do you mean with the brackets and the y? Are we taking an expectation here over the y's, or what's going on?
Yeah, so this is just an approximation, and I mean, by approximation, I mean, so you can think about the first line where this grad fx can be considered as difference between two neighboring points, and if you divide it by f of x, then that will give you an approximation of the term you have in the right side. So
okay, so you're taking the first line, you're replacing it with your density Q, and then you now integrate over all y. Is that right?
Yeah. So I mean, you basically, I mean, you don't integrate, but you actually calculate. You actually want to calculate the score that can I mean, point you to some neighbor which has higher mass in your discrete space.
Okay. Okay. What do you mean with the brackets? The brackets and the y at the bottom.
Oh, these brackets. Okay, so these brackets. Okay, so this for a particular point x. You can consider all the neighboring points and those. I mean.
Okay. So like integrating over all the y, right?
Yeah. So yeah. I mean, kind of. So actually, you want to calculate all the different scores and decide which direction you want to go. So basically, you will consider all the neighboring points, and then you calculate the score for all these neighboring points separately.
Oh yeah. Oh yeah. Yeah. Thank you.
Okay.
We have a vector here. You're just indicating this vector at the point x for all y or the Okay. Thanks.
Okay. Yeah. So I guess I have already explained this. And okay, so this is an example for a text where one Hamming distance our neighbors can be, and and for a DNA sequence of length 4, we show the possible density ratios we want to model with our neural network S theta x. So this is just one example. Okay. So with the goal of learning positive probabilistic quantities with neural network S theta of x, the convex loss function was proposed that correctly accounts for the positivity of S theta of x. There are previous approaches that tried to achieve the same goal with L2 loss, but here we consider Bregman divergence with the negative entropy function, and that's why this is called score entropy. And here is a proof that shows the minimum point reaches when S theta of x matches the ratio. In practice, the score entropy loss for all values of x can be combined using QX that allows us to approximate it with Monte Carlo sampling. In practice, score entropy is intractable, which can be transformed to a tractable equivalent following some standard techniques from score matching. Basically, we consider any kernel and corresponding transition to a sequence X from an initial sequence X0. This allows the density ratio to transform to another ratio that can be calculated easily. Okay. Now we have a tractable version of the loss, which is called denoising score entropy. Yeah. And so now we summarize the problem we formulated before, that is learn a neural network to approximate density ratios in a constrained scenario. We showed the loss function used to train the network where the ratios can be easily calculated with the transition rate matrix, RT. So let's revisit the loss function and the tools to calculate them. We have already seen the transition equation and the condition that the transition matrix should be considered in such a way that Qt tends to a terminal distribution for large T. We can use this transition equation to calculate the loss and train our model. But computing columns of exponential beta T RB is difficult for every T with the noise level beta. So here specifically, we follow the discrete diffusion work and decide a special structure for transition matrix as shown here. And Hans, so this is the specific matrix we use at the right side. This structured matrix perturbs the input DNA sequence according to the uniformly sampled time T, with all the matrix entries 10 to 1/4 for a large T. Yeah. So here we show the complete training process where, starting from a ground truth sequence, we can derive a sequence at any uniformly sampled time step T following the standard equation. But we actually allow our choice for our transition kernel as a spatial structured transition matrix that we saw in the last slide. Then we can calculate all the density ratios for sequences with one Hamming distance away. With that, we calculate the denoising score entropy loss, which in turn trains our network S theta of X. Please note that the size of the density ratio matrix, that is output size of the network S theta of X, should be D cross N, where D is the number of tokens, which is 4 for our case, that is A, C, G, and T, and N is the length of the sequence. After the training is done, we expect our network to learn the density ratios for the constraint scenario we considered here. This formulation allows our network to accept a conditioning signal, scalar or vector, alongside the data as input to the score network, which learns the density ratios based on the provided information. This approach differs from classifier-free guidance, as the conditioning signals consist of continuous valued scores, aligning precisely with the formulation for conditional training. Previous methods such as Dirichlet diffusion score model employed this type of conditioning for designing human promoters. And here we train our score network considering both transformer and convolutional networks. We used the same convolutional architecture containing 20 layers to compare the results with the previous approaches. And the specific transformer architecture we follow has 12 transformer blocks, 12 attention heads per block, and uses flash attention. And we get a better result with transformer architecture than convolution, which will show later in detail. Yeah.
Okay, then.
Yeah.
Okay. You're now talking about inference. Good. Let's go.
Yeah. So yeah, I mean, I'll talk about inference now. So we have seen the forward process where QT approaches some noise distribution Q base, which is easy to sample from, such as a random value in each position. The reverse diffusion process can be derived that reverses the density from Qt or Qbase to Q0 or Q data, as shown here with the reverse transition matrix R bar T. And now we have now the reverse transition matrix again contains a density ratio, and this time we are in luck. These density ratios are nothing but the concrete scores that can be easily calculated with the trained network ST-tags. And this helps to reconstruct the transition strategy in reverse, which intuitively pushes towards higher probability mass region. Yeah. So we just replace the reverse transition matrix with containing S theta. And this is the sampling process diagram where we sample from the terminal distribution. Just a reminder that we have a random matrix as our terminal distribution where each nucleotide is equally probable. Then we construct the transition densities for the backward steps with the backward transition strategy. As we need our intermediate sequences to follow a distribution, we normalize every column to 1 and sample each nucleotide at each position. We apply the backward step on this intermediate sequence again and continue until we reach at the final step at t0 0. And as our score network learns ratios based on some conditioning signals, it also accepts a condition during sampling, and the network drives the generation based on that condition.
Okay. So to assess the quality of the generated sequences, we came up with a wide variety of in silico evaluations. And these evaluations span functional similarity, sequence similarity, compositional similarity, informativeness, and capabilities for conditional sampling. So for functional similarity, we want to assess whether the generated synthetic sequences exhibit the same distribution of functional activities as the natural genomic sequences. So similar to previous studies, we use the conditional generation fidelity. This uses the supervised oracle to provide functional labels for the sequences. So here we generate synthetic sequences conditioned on the activity of matched genomic sequences. We get labels for each sequence and then calculate the mean squared error. The Fréchet distance is similar, but instead of predictions, it measures the probability of sampling the synthetic sequences according to a multivariate Gaussian model of the penultimate representations from a supervised oracle. And another way to view this is through the predictive distribution shift, which measures the largest gap in the cumulative distribution function of functional labels given by a supervised oracle again. And we do this for matched genomic and synthetic sequences, matched by activities. And the measure is essentially the Kolmogorov-Smirnov test statistic. To assess sequence similarity between synthetic and genomic sequences, we have 3 different methods here. Percent identity measures the extent of memorization as well as the diversity of synthetic sequences. It's a pretty weak similarity measure for regulatory genomics, but it's a straightforward test of exact memorization, so we included it. The Kamer spectrum shift is more suitable for regulatory sequences. Here, we calculate the Jen and Shannon distance to evaluate similarity in Kamer distributions. Lower divergence indicates a higher similarity between the synthetic and genomic sequences. So we'd like to see low percent identity and a lower Kamer spectrum shift, which indicates similar Kamers within the regulatory DNA, but not through explicit memorization.
Okay, how do you construct this? So this Kamer spectrum shift is you have a distribution of Kamers in your whole data set, and the distribution of Kamers in everything that you generated, and you want the difference between those two empirical distributions to be low.
Yeah.
Okay. that's the, like a distribution, checking that your distribution that you generate is right compared
Yeah.
to input distribution. Why does it? Why do you put that under sequence similarity and not under like distribution matching evaluation or so?
Yeah, I mean, it's just I guess it's not explicitly a sequence. It's whether the sequences. I guess it could have been put under like compositional similarity, because in regulatory DNA, the protein binding sites motifs are typically a k-mer size, and here we're just testing whether the sequences exhibit some sort of covariate shift, but not at the nucleotide level, at some k-mer level.
But I mean, in the percent identity metric, if we if we spit out the data set again, then we do poorly. In the k-mer spectrum shift, if we spit out the data set
Yeah.
again, then we do well.
Yeah. So this is why one single metric isn't sufficient. And the way I like to think about it is you want k-mer spectrum shift to be low, but you also want the percent identity to be low too. So you need to consider both simultaneously, or else you're never gonna really like you're basically never gonna come up with a good metric that satisfies all your conditions.
I would see the k-mer spectrum shift as a sort of yeah, a similar metric to the FID, right? We're also comparing distributional similarity.
Yeah, I can see that too.
Anyway, then
Oh yeah,
discriminatability.
discriminatability. This is a really hard word to say. Uh, so this assesses how well supervised models can distinguish between synthetic and genomic sequences. So this method is analogous to a GAN discriminator, but applied in a post hoc manner. It helps to identify any obvious covariate shifts that a discriminative model could easily exploit. Okay, so compositional similarity aims to evaluate whether synthetic sequences contain key elements of regulatory DNA, particularly motifs and motif grammars. And so we employed several methods to assess compositional similarity. First, we conduct a standard motif enrichment analysis using a database of known motifs and examine their co-occurrences as well. And while this approach is valuable, it has several limitations. It only considers known motifs from databases that we know to be incomplete, and motif scanning is also known to lead to many false positives. And moreover, the motifs that are being scanned are not directly linked to function. They're just observational, pattern matching. So we also analyze functional motifs identified through attribution analysis, such as gradient CHAP. And this method allows us to assess the composition of functional motifs now, at least through the lens of the supervised model. And it also allows us to identify patterns that aren't necessarily present in existing motif databases as well. And furthermore, we've developed an attribution consistency metric to quantify the consistency of patterns within a population of attribution maps. And this helps us to assess the compositional consistency of the functional motifs within the generated sequences. So first, we benchmark D3 using an established data set for diffusion models, which is based on the conditional generation of human promoter sequences. And so the conditioning is based on K-seq data, an experimental technique that measures transcription start site activity, which serves as a proxy for expression levels. Here, the sequences are 1 kb long, and the K-seq track is also 1 kb long. And this data set allows us to assess how well the diffusion models can conditionally generate synthetic promoter sequences with desired expression levels. In this benchmark, previous diffusion models have been evaluated using the conditional generation fidelity. Again, this metric involves generating a sequence based on a conditional value, and then using an oracle to assess the functional fidelity of that sequence. The performance is measured by the mean squared error, where a lower value indicates that the synthetic sequences better matches the functional activity of the natural sequence. And the original study was Dirichlet diffusion score matching, DDSM, and they incorporated other approaches, including bit diffusion and D3PM. And more recently, Dirichlet flow matching improved upon DDSM. So here we further expanded the benchmark using D3 models, which is our DNA discrete diffusion approach with this score entropy. Specifically, we compared D3 models using two different base architectures, a CNN and a transformer. And this was the same CNN used by the previous methods, which enables for a fair comparison. And the transformer was included to see if we could do even better using a more expressive model. And for comparison, we also trained Dirichlet flow matching DFM using the same transformer base model as well. In addition, we include a random sequence baseline. And so our results demonstrate that D3 with transformers produces sequences that are not only diverse, but also functionally similar to natural promoters with a lower MSE and a smaller predictive distribution shift. And these synthetic sequences also exhibit motif distributions that closely resemble those found in natural promoters, but the sequences are not being memorized, as indicated by the lower percent identity, or similar percent identity as even random sequences. This suggests that D3 effectively captures the complex patterns of complex patterns and functional characteristics, of human promoters, at least through the lens of the supervised oracle.
Can we maybe go through these
Sure.
columns, and just out of personal curiosity, look at the ones where the flow matching does super bad, and like even worse than shuffle sometimes, right? And oh yeah, I mean, okay, no, it kind of makes sense that it does worse than shuffle for some of these. Where shuffle means that we just take original sequences of DNA and or original promoter sequences and shuffle them, and then we check if these statistics are still right.
Yeah.
Okay, and then the motif co-occurrence. Oh no, the Skamer spectrum shift, pretty clear. The Lie flow matching doesn't have the right K-mers that you usually observe in DNA. And then also if we look at percent identity, I guess, okay, no, that's fine. Then motif co-occurrence, what was that again?
This is doing motif scanning and then just creating a vector for every sequence for all the motifs that appeared, and then just calculating a covariance matrix. And we do the covariance matrix for the synthetic sequences as well as the natural sequences and just calculate the Frobenius norm.
Okay, yeah. Okay, good. Very nice. Thanks. Okay, now then let's linger on this this evaluation as well. On the first column, what do you actually think about the first column? Because you know some of the biology here also a lot better. Right, we are predicting the or this I have no idea what this no idea, but there's this model that predicts a lot of numbers for an input sequence, and then we take all of these numbers and then we average all of the numbers and then we take the MSE between the average. So do you I mean, I can just ask, right, there seems to be a correlation, or at least models, there seems to be a clear, significant difference between the models. It's not just a tiny difference. Like if we rerun these models, we see that there's a significant difference. And it also correlates when you do better in terms of these maybe more relevant metrics on the right, and you do better in this MSE metric. So yeah, do you have a notion of what are we evaluating with this MSE? And
so in this case, it's not like a really great Oracle model, in my opinion. It uses the say model, which is trained on lots of different tracks across different organisms and stuff. And we're using H3K20 histone mark as a proxy for our Oracle for this KC model, for this KC, Oracle. it doesn't make too much sense that that's the right oracle. And so like we're going to move on to fly enhancers where we had more control over what our oracles are. And so, you know, I wouldn't linger too much on this particular task. And I'm going to also highlight a lot of major limitations of our evaluations period that we're relying on these in silico oracles. And if generative models are actually generating or learning a lot better cis-regulatory codes, you know, evaluating using in silico oracles is really limiting for us for our evaluations. And so I'll circle back to that at the end of the presentation or at the end of this. Okay, so next we introduced a new benchmark based on a fly enhancer dataset, which offers a different perspective on regulatory genomics. And this dataset is based on the StarSeq assay, a method that quantifies enhancer activity now with a single scalar value. It consists of, the dataset consists of 240 nucleotide long sequences, each associated with 2 scalar enhancer activity values corresponding to 2 different conditions, one for a developmental promoter and another for a housekeeping promoter. And the original study that produced this dataset used a convolutional neural network, or CNN, to fit this dataset using a multitask regression setting. And building on this foundation, we trained both our D3 models and Dirichlet flow matching models using the same 2 base architectures, the CNN and the transformer. And in our approach, we simultaneously conditioned the models on both activity values. Okay, so our evaluation of the fly enhancer dataset yielded consistent results with our previous findings for the human promoters. D3 demonstrated pretty good performance across all the evaluation metrics we tested. The D3 generated sequences exhibited higher efficacy in their function. They exhibited the same composition as natural sequences in terms of motifs, and even a downstream discriminative model could not tell them apart, as indicated by an AU-ROC close to 0.5 in the discriminability test. So this reinforces the effectiveness of D3 in capturing the generative or generating regulatory genomic sequences. Okay, so next we investigated how informative the synthetic sequences are. And by informative, we mean whether these sequences provide additional information that can benefit a supervised DNN as a data augmentation, for instance. So our hypothesis is that if synthetic sequences are accurately capture cis-regulatory codes while providing more genetic diversity, they could enhance the DNN's ability to learn and generalize. And so to test this, we conducted an experiment using the DeepStar dataset. We systematically reduced the size of the training set and then added synthetic sequences generated by different diffusion models. And it's important to note that the diffusion models were trained using the full training set, and it generated the same number of synthetic sequences as the full training set. So while the natural training set, which is plotted along the x-axis, is varied, the augmentation was provided, was the same size as the original training set. So while this isn't a realistic scenario for actual data augmentations, it serves as a useful test for whether the synthetic sequences contain any added information. And so the results are encouraging here. Even when using the full training set, adding synthetic sequences from D3 improved the performance of a freshly trained DeepStar model. And so this suggests that D3 synthetic sequences provide greater genetic variability while still encoding meaningful cis-regulatory information. And moreover, the benefits of this augmentation with D3 generated synthetic sequences were more pronounced in the small training data regime. Okay, next, we investigated the model's potential to generate sequences with task-specific activity levels. And this capability is important as it shows the model's comprehension of the underlying biological mechanisms and opens up a myriad of practical applications in synthetic biology. So specifically, we compare 2 models, DFM-conv and D3 trans, we tested their ability to generate sequences with predetermined activity levels for two different tasks, developmental and housekeeping promoter activities. In the top row, we look at cases where both activities were set to the same levels, high, medium, and low. And the first number represents the activity of the developmental task, and the second is a housekeeping task. And the top row tests if the models can generate sequences that yield similar functional levels in both tasks. And the middle and bottom rows examine a different scenario, generating sequences with high activity in one task, but low activity in the other. And we used the supervised oracle, this time using the DeepStar model, which was trained on the full training set of the StarSeq dataset to evaluate the generated sequences and predict their functional activities. And the results show that D3 performs quite well at this task. It excels at generating sequences that closely match intended activity levels, irrespective of whether the activities are similar or different between tasks. And so this indicates that D3 has captured meaningful biological patterns and can apply them to create synthetic sequences with desirable properties. Such a capability could be instrumental in designing synthetic promoters or understanding how different regulatory elements contribute to gene expression. And so in the study, we extended score entropy discrete diffusion to DNA sequences and performed extended evaluations of the generated sequences. We showed that D3 appears to have made significant strides towards generating synthetic sequences that appear to have the same characteristics as natural genomic sequences. However, while we have introduced a lot of new evaluations, in silico evaluations, it would be a shame not to highlight that they're not perfect and there are several limitations. For instance, many of our evaluations rely on in silico oracles to provide labels of generated sequences. And this just evaluates how well the generative models align with the supervised oracles. If the generative models learn cis-regulatory codes that are better than the supervised oracles, this cannot be properly assessed with using these supervised oracles. The only real way to do benchmarks of supervised models and generative models is through new experimental measurements, but this is, of course, quite costly in terms of time and labor. And my lab is strictly a dry lab, which without such capabilities. So if any experimentalists in the audience, in industry or academia is interested in helping us advance AI for genomics, we'd love to collaborate. And another limitation is that we are largely interpreting the cis-regulatory codes from the synthetic sequences through either existing bioinformatics tools like motif analysis or attribution analysis using a supervised oracle again. If the generative model is learning better or more expansive cis-regulatory codes that the supervised oracle is missing, we can't properly assess this. We desperately need new tools to directly interpret cis-regulatory codes from these generative models. However, such tools are not yet established, and so my lab is working towards closing this gap, hopefully soon. Another limitation is that biology often lives in a small data regime, and it's unclear to what extent these diffusion models can work in such regimes. And so we're actively exploring this as well. And overall, I'm quite excited and bullish about diffusion-based generative models for DNA. Ultimately, we're not married to what we're proposing here, the score entropy discrete diffusion D3 model. We don't really care about which specific modeling approach ends up performing better than others. Everything gets beat out in time. But what we do care about is advancing this field so that we can make real progress towards improving human health. And so if anyone is interested in working together on this endeavor, we're more than happy to benchmark your methods, brainstorm co-application ideas, or collaborate to push boundaries of generative AI for DNA sequences. And moving forward, we have several open questions that I think we as a field need to really tune and figure out. For instance, what benefits, if any, will generative models for DNA provide over the more prevalent supervised models that have already been proven to be quite useful? And what new applications can generative DNA models help accelerate? And we don't necessarily have good answers for these open questions yet. So if anyone in the audience has any good ideas, I would love to hear what you think. And with that, I'd like to thank other people besides me and Anirban who helped us advance this project, including Amber and Chris, who was a rotation student in my lab at the time, who helped us develop some of the evaluations. And we're happy to take any additional questions.
Very nice. Thanks for the presentation. And yeah, what do you imagine, or if you have already something cooking, what are these improved evaluations that you're thinking about for closing the gap? If you anything that you can say, if you can't say anything, that's it.
Yeah. I'll just say a few things. In terms of like seeing how good these models are generating, or like because ultimately we need to compare them against supervised models. All these relative comparisons against other generative models is good for the field right now, just to see how we're doing. But it's supervised models in genomics are so widespread. This is the standard that you have to be and you have to demonstrate that you're doing something more useful than supervised models. And there's no way we can evaluate this without doing actual experiments. And so we're trying to collaborate with a bunch of different labs right now, experimental labs that can easily generate, or that I shouldn't say easily, that are willing to help us evaluate some of these generated sequences. And so that is the number one thing. So getting actual experimental measurements and doing real benchmarking. And in terms of model interpretability, we have ideas on trying to understand based on like our conditional sampling, where we can like freeze some things, like we can introduce motifs and see what other what the generated sequences are, so that we can understand rules of the cis-regulatory code that the diffusion model might learn. And those are the avenues that we think we're going to go down in terms of model interpretation.
And in DDD, is there anything different than in SET, or am I missing something there?
DDD, can you repeat that?
In the, you call it D3.
Yeah.
Is D3 anything different from SET in terms of the discrete diffusion framework or not? Can I just read the SET paper to understand everything that's going on there?
Oh, you mean the original D3 paper, or the original score entropy paper?
Yeah.
Yeah. So
yeah, I mean we have just modified the original score entropy to make it work for DNA sequences, and made changes to the matrix and that I already showed. But apart from that, the overall framework is very similar.
Yeah.
Good. Denise, what's your question?
Yeah, I just wanted to ask a couple of questions, conceptual ones, I think. Well, the first is very naive. It's about sequence modeling. So, do you have fixed size sequences, or can you expand your sequence after training and model arbitrary length sequences?
This depends a lot on the architecture of the models, right? Our CNNs typically,
I think it doesn't.
I don't think it matters, right? But
yeah,
I'm trying to remember the architecture of the CNN, if there's any like flattening or something, or any pooling. I don't think there was, right? There wasn't.
Actually, there is. I mean, you can actually generate arbitrary length sequence.
Yeah.
So I mean, the way the networks work, you can actually generate.
But I don't know what the capability, like how good the quality would be if you were to just scale up. But that's something that needs to be evaluated.
So Denise, so there's, as far as I understand, nothing depends on the architecture. But you cannot, like in your language model, right? You can predict a stub token, and then you have generated a sequence of that length. But here you have to decide before you start your diffusion how long your sequence should be. So you could sample that number of nucleotides, for example, from a categorical distribution.
Yeah.
Yes,
Yeah,
yes,
but although
exactly.
one can do that, it's not clear that you're going to generate anything meaningful if you scale up way too much.
Yeah.
Mhm. So what is slightly confusing to me is that your transition or rate matrix is evidently not spatial, right? It only considers a single nucleotide, so it's essentially 4 by 4. Or am I missing something? Because in the paper you say like it's dependent on D, but I actually thought that it was a fixed length size. But
Yes. I mean, actually, the transition matrix is 4 by 4 because we have 4 tokens, and if your token size is n, then the matrix size should be n cross n. So yeah, it depends on the number of
okay,
tokens you have.
yeah. That's what's confusing, because if you have a fixed size transition matrix, then you must reapply it along the sequence somehow, and I don't understand how it is done.
Okay,
Mm,
so the transition matrix, the reverse transition matrix changes across the sequence, right? Your
Yeah.
reverse rate matrix stays the same across the sequence. Can you maybe go to the slide where you show the reverse transition matrix? Because the reverse transition matrix, I don't think it's here. Yeah, the reverse transition matrix, where we have the arrow, right? Arrow, no, yeah, here, for example, right at the bottom we have the reverse transition matrix, and it includes this the score, and the score is learned by our neural network, and our neural network takes all the positions of your DNA as input and produces for this one position in your DNA this I to J value, and or like the vector of one I to all the J, or one J to all the I don't know. And then from that you obtain the the reverse rate matrix, and from the reverse rate matrix you obtain the reverse transition matrix.
So if I understand correctly, then you consider a context of some fixed size length, but you output on each iteration, you output only a single nucleotide, so you can slide it.
Nope, I don't think
Nope.
that. I think your neural network takes in all of the sequence, right? And then for every position in the sequence, it gives you 4 values, which is your score. And then this score you multiply with the with the transition, or with the rate matrix, to get your reverse rate matrix.
Yes.
And
Okay.
then you have this reverse rate matrix that is different for every single one of your positions. And then you apply it to every single one of your positions. You get out some probabilities, and then you sample these probabilities, and that is then your new sequence. And then you repeat, and repeat these reverse diffusion steps, or these denoising steps, and end up with your final sequence. Correct.
Okay.
Yeah. Perfect. Yeah.
Yeah. Okay. But the scaling key is quadratic then, or not? That's also what is
in sequence length.
Yes, in sequence
Scaling
length.
in sequence length, that depends on your architecture. And if you use a CNN, right, then it will not be quadratic. And yes, then you're in the situation where you only see a window around your around
Yeah,
the.
I was more asking about the transition matrix because it's seemingly squared in length.
Right. I mean, no, not in sequence length. It doesn't depend on sequence length, right? The size of your rate matrix only depends on the size of your vocabulary. And if you do it on language, like they do in the original set paper, then it's a 50,000 by 50,000 matrix, and
Okay,
the
okay, yeah. If that's the case, then no problem. Yeah, I was confused
So
by.
So basically, I mean, by applying this reverse transition matrix, you sample for every position, and for every position, you need to sample for. You need to have like 4 different values, and that depend. That's why that depends on the size of the matrix, that is 4 by 4. And if it is 50,000 length, 50,000 tokens for our text modeling, then you have like 50,000 by 50,000 matrix
I guess.
will be used. But the sampling is for every single position of your sequence, and it doesn't depend on the length of the sequence.
Okay, I see. And the last question is just how does it, like, what is the main advantage compared to other sequence modeling approaches like DNA
Right.
and so
I
on?
also had that question. I suppose maybe in your lab you've had more experience with DNA generative models, and I assume you've also messed around with like language model approaches like hyena DNA or so in the past. Does it seem like diffusion models are doing better than language models? And you also said this, you're quite
Yeah.
bullish on diffusion models. So yeah, how comes? Any ideas?
Yeah. It's because I think it's just the objective function really. Any reconstruction-based losses, if you have vast majority is just junk DNA, it's hard for models to just learn or to predict that. You're like equally weighting uninformative positions as just as important as informative positions in your losses, right? Even with matched language modeling or even next token prediction like causal language modeling. And if the vast majority is uninformative, you just have this really nasty high entropy of predicting nothing, predicting garbage. So it's like if you had a language model for proteins and you just injected random amino acids between every important amino acid and of variable lengths, would a protein language model still work? I think it'd be really hard for protein language models. And that's the regime that we're working with DNA. So we actually have another preprint where we benchmarked hyena DNA, nucleotide transformer, and a bunch of other language models that were trained on whole genomes, and we evaluated them on non-coding DNA regulatory genomics tasks. And we show that all these models really struggle quite a bit in these areas.
Okay.
Yeah.
Interesting. Good. Then, Denise, if that also satisfies you, then I would move on to Yuning. What's your question?
Right. Hello, Anirban and Peter. I have a question. Do you think it is biological true that the regulatory sequence are downstream sequence specific, which means different downstream sequence probably require different, for example, enhancer or promoter sequence?
I'm
Like
sorry, I don't
the
really follow the
protein,
question.
is it specific to the protein that we're trying to enhance or promote? Is that the question?
Yeah.
Like,
Oh,
are you
I see. So are you asking like whether a regulatory sequence is dependent on what they're trying to regulate?
Yeah.
So yeah, I mean, to an extent, right? Sometimes they could have that. There could be enhancers within the coding regions, for instance, as well. There are notions that there are, strong promoters and strong and weak promoters as well. And so like that is more of an intrinsic property of that, right? Irrespective of what it's promoting. And enhancers interacting with promoters they have been found to exhibit compatibility rules that we don't really fully understand. So sometimes enhancers could be next to a promoter, but not actually enhance it. It might skip over it and enhance another promoter down the road. So all of these things are really just open questions. And I think in biology, it's hard to make any definitive statements about how biology works in general.
I see. Yeah, I just see in your conditional generation scenario, the conditions here is only the activity represented by some scalars number.
That's
So
right.
I'm wondering whether that question is true and whether we can add the downstream sequence into the downstream nucleotide sequence into the conditions as input to the model.
Yeah, that's a great point. It's something that we haven't explored, and we haven't really thought too much about it since this whole area is relatively new. I think, yeah, we don't know.
Okay, thanks.
Okay. Hassan?
Hi, yes, I just had a question. Have you tested the models against the Bend benchmark?
What
Can
do
you
you mean?
repeat that?
What then?
The Bend benchmark. So that is the one out of Copenhagen, the standard paper. So they did, they test the models by looking at the embeddings and seeing if those embeddings have actually learnt important features of the task. So they do gene finding. Hold on, let me bring it up.
Okay, so I
I've
guess
linked
there's
it in the
a benchmark called Bend, B-E-N.
Oh yeah, Bend. Yeah. I'm familiar with that. Yeah.
What
So,
a, yeah.
so was the question, have we thought about doing Bend?
Yeah, so have you tested your models against Bend? So have you tested D3 against Bend?
No, we haven't. And we're only specifically focusing on regulatory sequences in our training of these D3 models. It's not genome wide. So we need to be able to scale these diffusion models up to encompassing much larger DNA sequences. And I think then we can start doing genome wide training them on like whole genomes, for instance, like what people are doing with language models. And then it might understand notions of coding regions and non-coding regions and so forth. But that hasn't been explored. We're in our study and in previous diffusion models for DNA, we've been only focused on specific types of regulatory elements. So Bend benchmark wouldn't be appropriate.
But you can test the model itself against Bend. So they have like a section that's just for the fine tuning, and then you can compare different architectures. So they use the ResNet, CNN, and in this instance it would be a D3, and then
But I
So
think
just seeing if
it would only be appropriate for maybe a subset of the Bend tasks.
Yeah. So for the gene finding and a few others, you'd be able to do it.
Yeah, maybe a few, but yeah. It's something to explore for sure.
Yeah. Okay, thank you.
Okay.
Yeah, I still wanted to ask last one question because I actually was going to ask it before about scaling. So because in my view, diffusion models, the main advantage of those compared to sequence modeling is that they can be scaled in a more hierarchical way, maybe. And the idea is that you can apply like hierarchical diffusion and also output whole sequences at once. But in your approach right now, you output basically one nucleotide at once, and I don't quite understand how this can be scaled to the level of the whole genome. So you probably would need to output whole patterns.
Exactly. So this method definitely will not be able to scale up to very, very long sequences right now. And we're trying to come up with strategies on how to scale it up, but it wouldn't be using this one Hamming distance away kind of diffusion walk, because that we just couldn't do that. So we have other strategies in the background right now that we're trying to explore.